Okay, thanks for the invitation to participate of this nice event and for sharing with you our results of some papers that have been published recently. So the name of this lecture is Searching for the Best Way to Deliver Probiotics to the Gut. So I'll start with some good news that some of our best friends are germs that live inside and outside our body. And the Human Genome Project opened the door and made it possible to access the sequencing of the gut microbiota. And new scientific discoveries have pointed to deep connections between our health and the microbiome. And not just scientists got interested in this, about these connections related to our health and the microbiota, and also the modulation of the microbiota by probiotics and prebiotics, but also general population got interested, and especially in some situations where we identify signs of imbalance of this community, such as during long-term diarrhea and constipation. So in these situations, normally people go to a drugstore or to a supermarket and buy products containing probiotics and prebiotics. So we have some options to buy, and they can be food products, fermented or not fermented products, or even supplements like capsule tablets and so on. And this market, the global market, is growing and getting profits, as you can see here, some numbers. So let's imagine that we are in a market where we have so many options as in this picture for probiotic products. So let's imagine that you have a problem, diarrhea, for example, and you come here and choose a product here to help yourself. Okay, let's imagine that you, just to make it simple, let's make this example just to face it, that you choose a supplement with a single culture, with just one strain. And the scientific name of this strain is on the label, is declared on the label of this product, okay? So, but let's imagine that you also find products there in the same supermarket, can be a dairy product or a dessert or a beverage with the same strain, exactly the same microorganism, probiotic microorganism there. So is it the same to take the probiotics from the supplement or the food? This is an unsolved question till now. So let's try to draw a conclusion about this question, to find an answer. So let's imagine that we took these two products and to a lab, a microbiology lab, and we analyze the quantity of the viable cells. And let's imagine that we got the same counts of these microorganisms. So it was the same strain and the same count. So when we can conclude that apparently there is an equivalence between taking these probiotics from food or from the dietary supplement, isn't it? But we need to do further comparative studies, not just plate count in these conditions are enough to draw a conclusion. And this kind of information is important to consumers to make them, to give them information to the best cost-beneficial relation, and also for scientific and regulation purposes as well. So with the same example, I propose to you, if this product we chose at the supermarket was an yogurt. So in this product, yogurt is a liquid or almost liquid product. So we have high water activity, and these cells there are metabolic active. It means that they are replicating, they can replicate, they can produce metabolites like organic acids. They are viable and culturable. For sure, if you put in a stressful condition, like in your stomach, they can be injured, they can be irreparably damaged, and they can even die. But making the comparison with the supplement, they are not in a metabolic active state. They are in a dormant phase because we have a dry product. So they are not active there. They are in a dormant phase. For sure, if you put them in a nice condition, like put in water, give time, they can wake up. But if you put in stressful condition, like in your stomach, and your stomach is not a friendly environment for these microorganisms, they can just die. So just plate count is not enough. We need first do simulate the digestion and then plate count. And we can use some tools like flow cytometry, so we can check metabolic activity, cellular integrity of these cells, and also we can use microscopy to see the morphology of the cells. So I will present two papers that we published. The first one was published last year at the Journal of Functional Foods. So this is the graphical abstract of this paper. And we bought at a drugstore a dietary supplement, which is composed by 2 probiotic strains. The strains were declared in the label, and also the quantity of them, and also inulin. So we first analyzed this product, inulin, by chromatography, and the probiotics plate counting in selective conditions. And then we made 2 food products, 2 dairy products, an yogurt, not an yogurt, a fermented milk, and an ice cream. To do the fermented milk, we used no starter cultures, no Streptococcus thermophilus, for example. Just these 2 microorganisms to ferment the product. And so we included the inulin. Then we analyzed it, we simulated the digestion of these products at the same conditions. And further, we did the analysis of the survival, plate counting, the metabolic status by flow cytometry, and morphology by scan electron microscopy. Here I cover the results to not spoil the presentation and keep you interested. So I think this is pretty clear, but the aim of our study was to analyze to compare the survival, metabolic status, and morphology of the probiotic cells after simulated digestion in these 3 possibilities of taking the probiotics: the dietary supplement, the fermented milk, and the ice cream. 3 of these products contain the same strains, the same quantity of them, and inulin as a prebiotic, so we got a symbiotic product there. 3 symbiotic products. So first results. As I told you, we use selective conditions to count the 2 probiotics declared there, Lactobacillus acidophilus and Bifido animalis, and we obtained these counts: 9 log cycles for acidophilus and 10 log cycles for Bifido animalis. The quantity of inulin was 22.2%. So without the selective conditions, we obtained the total count of probiotics, and we made the 2 products with the same quantity, almost the same. The products were very similar, comparing, taking account the functional components there. So as you can see, they are pretty similar. So then we made the analysis of flow cytometry. So first, let me give you some information to make it more easy to see the results we got by this analysis. So in the left, we have samples before digestion. On the right, samples after digestion, the complete digestion, okay? And we use it for this analysis. Those are dot plots, is the way we take the results. And as you can see, we used two dyes. One of them was to stain the cells. We cannot see Acidophilus and Bifidobacterialis. We don't know what is each one. We see both of them here because they are very small cells, so it's not possible to separate what is one and what dots correspond to one and what dots correspond to the other. So we use these two dyes, as I was telling you. So CFDA is a dye that stains metabolic active cells. So here in this quadrant, we have live cells. On the other hand, PI is a dye that is not permeant to the membrane of the cell unless it has been damaged. So when it's damaged, the dye can go inside the cell. So here, when we have both dyes staining the cell, we have cells that we call damaged cells. They are alive, but they are damaged. And here, when CFDA is negative and PI positive, we have dead cells, okay? So, taking a first glance, we can see that before digestion, we got some cells alive from the three products, but probably higher quantity for the food products comparing to the supplement. Before digestion, we saw also that some damaged cells were there for the three products. Ice cream maybe for the freezing process, and dietary supplement for the drying process probably, but we can see high quantity even before digestion. And after digestion, we can see that the majority of the cells got killed by digestion. So the digestion process is very stressful for the cells. And this is the reason why we need to take high quantity of probiotics and constantly to get some of them, of the cells, alive. So here we have the same results, but with statistics. We can see here we made this graphic with live cells, damaged cells, and dead cells. And we can see supplement dark, fermented milk gray, light gray, and ice cream dark gray. Okay, so as we just saw, we got higher quantity of live cells before digestion for both food products compared to the supplement. This is before digestion started. Then we have gastric phase of digestion and enteric phase divided in two stages. So we saw that in the gastric phase, ice cream protected better the probiotics there, probably because of proteins and lipids they are in this matrix. But then at the end of the digestion, we got no further protection for ice cream, but we got higher protection of the cells for fermented milk, and this is statistically different from the others. Here is the comparison for damaged cells, as I told you, higher quantity of damaged cells for supplement, and dead cells, as I told you, the majority of the cells got killed by digestion, but we need to say also that even dead cells can have can promote some benefit, some health effect, and we call this the ghost probiotic concept, which is another subject to talk. Okay, so here we performed the plate count, and as we can see, we got a drop of 5 log cycles to the probiotics. Here the probiotics are together, okay, to compare with the flow cytometry, where we see them together, mixed. And we saw this drop of 5 log cycles for the supplement, and then more than 5 log cycles in the other phases of the digestion, steps of the digestion. For the fermented milk, we saw a drop also, but 3 log cycles only in the gastric phase, and then no more. They maintained the counts. And for ice cream, we got 3, 4 log cycles reduction, and then at the end, 5 log cycles reduction, similar to the gastric phase for the supplement. So in such a way, fermented milk better protected the cells, or the cells, this is my opinion, were more active to recover and to be there, even with the stressful situation imposed by the digestion. Here we got some pictures from microscopy. So let's start with the supplement. This first picture on the left is before digestion. We can see the cells here, very beautiful in my opinion. And then after enteric 1 phase, so we got gastric, and then we are in the enteric phase 1. And we can see that the cells got a different morphology. Look, they got this granular aspect. And probably this is because we got the membrane got damaged, and the intracellular content of these cells leaked out. So we got this different aspect, which is very, it's impressive because they got really, really different. Here we have the fermented milk, not just the cells, but we can see the matrix composed by this network of proteins, caseins and whey proteins, making this network, and this network protects the cells during the digestion. So as I told you, cells here are in a metabolic state. So here in the arrow, we can see that they are even replicating. On the right, we can see is the same phase of digestion we just saw for the supplement. We saw that some of the cells got damaged. We can see here the Y shape for bifidobacterium, and the cell was broken, probably by digestion as well, but we do not see that granular aspect that we just saw for the supplement. And here we have the ice cream. In this product, the cells, the probiotic cells, are very hidden, are very attached to the food matrix. So it's a little bit difficult to see the probiotics there. So I chose gastric phase and then enteric 2 to better see the cells there. So we can see here the matrix covering, protecting the probiotics. And then here we have the matrix almost digested. We are at the end of the digestion process. And we can see that the cells, some of them got a little bit granular aspect. Okay. So now I uncovered the results, and we concluded that the food products better protected the probiotics during digestion comparing to the dietary supplement. So the state of art on probiotic does not present a definite answer, but these results make us to believe that food products can better protect the cells. And even more, for fermented milk in this case. So now I will quickly present a second paper that we published at the British Journal of Nutrition. This publication was this year in 2020, and we did a next step. So here we choose just 2 products to compare, and as we have more studies with fermented milk, we choose to pay attention on ice cream, which is a matrix not with not so many studies. And we compared ice cream with the dietary supplement using a model called SHINE model. SHINE is simulated of the human intestinal microbiome ecosystem, as you can see in this picture. This equipment belongs to a colleague of mine called Katia Sivieri from Unesp, Araraquara, Brazil. And there are 2 advantages to use this equipment. Here we have a digestion that is continuous. And here we can put anaerobiose environment, and we can include microbiota. So this is very nice to do further analysis and further comparison. So we can include the microbiota you want here, and we chose from healthy people from the army, from volunteers from the army. So we took the stools of them, make a pool, and inoculated the equipment. Then we did a supplementation of 15 days with the dietary supplement in this equipment. We made a washout period, and then we supplemented them with the ice cream. And we analyzed the microbiota by next generation sequencing. We analyzed some of the metabolites, short-chain fatty acids, and ammonia. Short-chain fatty acids by chromatography, and ammonia we using an analyzer of ions. So here we got the results compared with the control period. The control period number 1 and control period 2, which we called washout. Okay, so as we can see here, for the supplement, we got a drop in ammonia, which is a nice result. Ammonia in high quantities can be toxic. So we got a nice result here. But we got a drop in some short-chain fatty acids, like acetate and propionate. But for sure, it was we got a nice result here, a rise in butyrate, but not statistically significant. For the supplementation with ice cream, we got better results related to the short-chain fatty acids because all of them were increased, and some of them statistically significantly, like butyrate and acetate. But ammonia, we got a rise. It was not statistically significant, but it's not so nice result. Then we analyzed by sequencing the microbiota, and here we are presenting the orders of the relative abundance of them. So we can, as we can see, here is the pattern of the order of the microbiota of these volunteers. And as you can see, control and washout have a similar pattern, which is very nice to our study, to see that the conditions were similar at the start of the run. And with the supplementation, we saw also a different pattern of distribution, of quantity of these orders, microbial orders. So we can see that both ways of supplementation can modulate the microbiota. And this is interesting. But we saw that for ice cream, we got higher quantity or lower quantity. It was statistically significant for some of them. I forgot to put the marks here, but I can show you that for lactobacillus, we got an increase, and which were more important. And also for Clostridialis, we got a drop more important. So we saw that ice cream modulated strongly the microbiota. And here we can confirm some things. The letter A is the principal coordinate analysis, where we can see that dietary supplement and ice cream clustered together, so they are related. And at the letter B of this figure, we can see the principal component analysis diagram. And as you can see here, ice cream clustered with the short chain fatty acids. So this is a very nice result that shows that probably cells, probiotics here, are more metabolically active to produce these important metabolites to our health. So both of the dietary supplement and ice cream treatments were able to modulate the microbiota. The supplementation with the probiotic ice cream showed to produce more quantity of short-chain fatty acids, but the dietary supplement were more efficient in reducing the ammonia content. And so we can conclude that each of the supplementation forms evaluated has advantages, could be considered as an alternative for probiotic consumption, and ice cream probably should continue to be an occasional foodstuff because it has high quantity of sugar and fat, so we need to pay attention on this as well. So we are finishing the third step of this research with a clinical trial. So next time I can tell you the results we got. Here is the picture, a nice picture of where I work, and my lab is almost here. And thanks to my team that work hard to get these results, especially to Vivian Rodriguez, and to the agencies that give us financial support. Thank you all for your attention. And I'll be glad to answer some questions. Thank you.
Thank you, Dr. Adriana, for the presentation. Oh, we have few questions in the chat box. So what I'll do, I'll just directly unmute the audience, and so that they can directly address their question to you. Okay, I'm going to unmute Dr. Joby. She has a question. I'm going to send Dr. Vinette Joby unmute request. Please accept it. Yes,
hello.
you can.
Hi.
Yeah, hi. This is Renita. So I wanted to know, what is the count of these probiotic organisms that you added before the fermentation?
Yes, as I showed you in the counts, we put the same quantity that we observed in the dietary supplement. So I don't have exactly the quantity of the powder because this information is with my student, but I can check this information. But we made some trials to catch this result. It was not the first time that we made it that we got this result. So we needed to try it many times to take this result.